So it's my honor to introduce Ismail Kangwehi, and my apologies, Ismail, if I mispronounced your second name. So Ismail's a final year PhD student in the Earth Science Department of Stellenbosch University in South Africa, and he's part of the Center for Trace Metal and Biogeochemical Cycles there. He was also a past participant on RGNO, which is where I've met Ismail before, and we really had a great time doing research together. So prior to his postgraduate studies, Ismail was a exploration geologist for a local company in Namibia, and he also completed his undergraduate degree at UNAM in geology and environmental biology. And so Ismail, as he introduced earlier, is going to talk about the impact of mineral dust from the Namib Desert for fertilizing the Southeast Atlantic Ocean. So over to you, Ismail.
So as I said, thank you for that introduction. Look, I'm just going to give a brief overview of one chapter of my PhD studies, and it focuses on the Namib Desert, where we look at the aerosols from the Namib Desert and how they can fertilize the East Atlantic Ocean as well as the Benguela system. Just to a brief introduction, dust is a key source of nutrients, and it can fertilize phytoplanktons. As most of the people in this group will know that phytoplanktons can absorb up to about 25% of all anthropogenic CO2. And some of the dust from Southern Africa is known to travel to some of the regions where some of these nutrients are limited, which can be supplied by the dust. Nutrients such as iron, such as cobalt, zinc, your phosphates and your nitrates. Dust can be a huge source of these nutrients. But over the last 20 to 30 years, there's been a growing interest, especially in dust research, because of global warming and the role that aerosols and dust can play in the removal of CO2 from the atmosphere. And when you look back into paleoclimatic reconstructions, there's quite a good correlation between aerosols, between when there was high dust in the atmosphere and a removal of CO2 from the atmosphere. When we get into Southern Africa, there's quite a lot of when you look at when you use remote sensing techniques and you look at satellite images, there's quite a lot of huge dust plumes that leave the Namib Desert that fertilizes the Atlantic Ocean. So part of my study was to sort of understand what does this dust do? Does it fertilize the ocean, and what impact does it have on the ocean? And one of the things that I really wanted to look to was the composition of the dust. The reason why we wanted to look at the composition is because the different sources in the Namib Desert have different composition of in terms of trace metal composition, and this plays a huge role. And more than the composition, we were also very interested in looking at the bioavailability of the dust. The reason we look at the bioavailability is because it's what actually gets available and becomes available to the phytoplanktons. So to do this, we calculated and we did leaching experiments. The reason why we did these leaching experiments is to sort of get the bioavailable fraction, and that's what becomes available to the phytoplankton. So we went out into the Namib Desert and we set out a lot of dust traps, and we set out stations where we recorded a lot of environmental parameters. We looked at the fog content in the atmosphere. We looked at other parameters such as the wind speed, the wind direction, to sort of understand where the dust travels to. And we also collected dust samples. And these are just examples of the instruments that we use to collect aerosols or dust from the Namib Desert. And we classified our different stations. I'm just going to talk about 4 of them in this talk, just because of time. Most of the studies tends to focus on what we call the dry riverbeds. But in this chapter, I focused on the gravel plains because they are less studied. And other studies have shown that they have high, the bulk composition of iron in these dry riverbeds is pretty high when you look at the background geology. So we investigated the soluble fraction from these stations, And these stations were classified based on location, and we classified them as the coastal stations, which were very close, like the coastal net, and the ones which were inland, which were, and both of them and all these stations had different bulk trace metal compositions, and their bioavailable fraction, which gets deposited into oceans, also differs based on those locations and the environmental parameters which are found in. One of the other parameters that we also looked at was the particle size distribution, because we really wanted to know the particle sizes at the different stations, because this gives us an idea and of how far the dust can travel, and it also gives us an idea of how soluble the dust can be. And as I've already stated, that the more soluble the dust is, the more bioavailable it becomes to the phytoplankton. So besides doing the bulk composition, we also looked at the particle size distributions. We looked at the geomorphology of the particles as well, because that gives us also an idea of how far the particles can travel. And we also looked at the mineralogy of the particles. And maybe what's worth mentioning was the fact that most of the particles in the coarse grain fraction, they were very similar, but the most interesting particles were really the very fine grain particles, the less than 50 micron particles, and most of them were found in aggregates. And what we found to be interesting about these aggregates was the fact that when we did the bioavailable fraction out of it, the bioavailable fraction was usually higher in these aggregates, in these because they are less exposed compared to when you find minerals which are individual grains. And just because of time, the bulk composition also showed that for example, when we looked at iron, the stations that had the highest that had high bioavailable fraction did not necessarily have the highest total trace metal composition, and that shows that there's, besides the total bulk composition, there's other factors that play a role, such as the environmental parameters that I spoke about earlier. And we also modeled where the dust travels to, and we realized that most of the south of the Namib Desert dust mainly fertilizes towards the Indian Ocean sector, and the Southern Ocean region, which is one of these high nutrient, low chlorophyll zone, is probably depleted in these trace metals, because most of the air masses pathways from the Namib Desert do not travel in that direction. On the flip side of things is that some of the dust can be toxic to the phytoplanktons, especially in high concentrations. When you have high concentrations of trace metals such as copper and cadmium, it can be toxic to the phytoplankton. We also know that dust can carry pathogens and microorganisms, so we really wanted to investigate some of these factors to see how the Namib Desert dust can impact the ocean. And just in short, that's my talk. And I also wanted to just say a word of thanks to Chibo, because she introduced me to the RGNO. She invited me to the RGNO, and I'm grateful. And may her soul rest in peace. Thank you so much.
Great. So Carl was a student in the 2018 RGNO course. And currently, Carl is a PhD student and a junior research fellow at the University of Tartu in Estonia. Carl is advised by Ivo Leplin, Lisa Lang, and Kalle Kursame. And since leaving the course, Carl has really been doing some exciting work on the origin of phosphorites and geochemistry of phosphorites. And Carl's work has been published in some really leading geology journals such as GCA. And I think today he's going to tell us about some of that work.
Yeah, thank you. First of all, rest in peace, Chibo. I must admit, I'm a bit shaken up by all of this. So if my presentation comes out less than perfect, sorry for that. But anyway, I'm going to talk to you about the formation of sedimentary phosphorites. The 7-minute format doesn't really allow me to go in too much detail. So I thought I'd give you a short overview of the general process and just sprinkle in some of my findings. But first of all, why even bother studying phosphorites? To paraphrase Isaac Asimov, in the future, we're probably able to substitute almost all material we use in our everyday life. But for phosphorus, there's neither substitute nor replacement, because phosphorus is a key building block of life. It's found from our DNA to ATP to the bones of mammals. And it's also a limiting micronutrient on a geological timescale, which essentially means that how much phosphorus is in the system dictates how much life can exist. So because of that, life has become quite efficient at taking up phosphorus, which means that very little of it actually makes it into the sediment. So the average sedimentary rock only contains 0.1% phosphorus, whereas in phosphorites, there's an order or two orders of a magnitude higher concentrations. As you can see on the map, there's only a few places on Earth where there is phosphorites being formed currently. So it's a rather peculiar sediment. And one of the few places that actually is active as far as phosphorites is concerned is Namibia. And on the Namibian shelf, the phosphorites are deposited as those like really patchy and irregular deposits. And the main apatite or the main phosphorus containing mineral in these sediments is apatite or calcium phosphate. As I showed you earlier, there are only a few places where phosphorus or phosphorites are formed, and because it's a rather complex mechanism how these sediments are deposited, and you need a very specific set of conditions to precipitate phosphorite. So there are three large prerequisites for phosphogenesis or the formation of phosphorites. The first main one is upwelling. So you essentially remove the surface layer of the ocean, and it's in turn compensated by the upwelling of deeper nutrient-rich water. And that nutrient-rich water is, or the phosphorus in that water is taken up by microorganisms. They use it to create more organic matter, and some of that organic matter is deposited on the seafloor. But even then, the concentrations are way too low to actually precipitate apatite. So you need another mechanism to pre-concentrate that phosphorus. And this job is done by a very unique group of bacteria called large sulfur bacteria, or LSB. So they're unique in that way that they have two different metabolic pathways. So usually they oxidize sulfate to sulfite to gain energy, but they also have a backup system. So let's say under suboxic conditions, or when there's some energy in the system, they gather up phosphorus from their surrounding environment and store it in their cells. But when things get tough, or oxygen or nitrate runs out, they metabolize that phosphorus and they release it into the surrounding pore water in short pulses. And that can drive up the concentration of phosphorus significantly higher, and then you can actually precipitate calcium phosphate or apatite. So this is like the general model of phosphogenesis. But there's still some unanswered questions, like, for example, in how deep of the sediment does this mechanism work, and what are the dominant biogeochemical conditions during this process. So what we found out is that if you look at the trace elemental composition of those apatite grains, and at the very center, the trace elemental composition is almost identical to seawater. So that essentially means that they have to precipitate at or near the seawater sediment interface. And also, if you look at the whole whole sequence of sediments, there's a very specific sequence. So you have organic rich sediments at the bottom, where there's a lot of sulfite, but not a lot of phosphorus or apatite. And as the system becomes more, let's say, high energy, then you start to precipitate apatite. So there's a very specific sequence of things that have to happen in order to apatite to form. Thank you.
Thank you, Caro.
Nicola is a past participant in RGNO, and currently she is a doctoral student in geobiology and organic geochemistry at Hamburg University, where she is currently studying microbe mineral interactions at hydrocarbon seeps. Nicola completed her master's degree at ETH Zurich with major in mineralogy and geochemistry, and today she will be showing us a little bit of her master thesis research, which is regarding the origin of the discrepancies between different sea surface temperature proxies. Nicola, the floor is yours.
Thank you very much, Blanca. So yeah, as Blanca already mentioned, I'm going to present some results of my master thesis, which was on the origin and distribution of organic matter in Southwest Iberian margin sediments and the implications they have for the sea surface temperature proxies. So various studies investigating past environments have shown discrepancies between different sea surface temperatures obtained from proxies within the same sediment layer. And further studies have also revealed that there are different 14C ages of the different components of these proxies, as well as 14C age differences of several thousand years, which is associated with different grain size fractions. So there's a possible spatio-temporal decoupling between proxy records, which raises questions about the geographic and temporal reliability of paleoenvironmental reconstructions. It is therefore important to understand the relationship of these discrepancies and to disentangle the reasons for these disparities by investigating the sources and distribution of organic matter with which these organic proxies are associated. So what we did for the study was we compared different SST estimates from surface sediment core tops from the Shackleton sites on the Southwest Iberian margin. And in addition, we investigated the spatial distribution of bulk characteristic of organic matter in order to gain an understanding of the origin and distribution of this organic matter. So as a bit of background information, I want you to know about the different SST proxies that we've looked at. There is the UK37 index, which is derived from alkanones, which are membrane lipids produced by coccolithophores, which live on the surface waters of the ocean. And these organisms alter the saturation state of their membrane lipids in order to maintain membrane fluidity at different temperatures. And the ratio of the C37.2 alkanone compared to the C37.2 plus C37.3 alkanone gives the UK37 index, which correlates with sea surface temperature. And the second ratio we looked at is the TEX86 index, which is derived from GGGTs. These are membrane lipids of archaea. And again, these organisms adjust the molecular structure by changing the number of rings within the GGGTs. And this also helps to maintain the fluidity of the membrane at different temperatures. And the TEX86 ratio is the ratio of the relative abundance of these cyclopentane rings. And the third proxy we looked at was the magnesium-calcium ratio of foraminifera, which are planktonic organisms with calcareous shells, and this ratio of magnesium within these shells also correlates beautifully with sea surface temperature. Then the second piece of information, which is important as background information, is that there is something that discrete grain size fractions are sorted hydrodynamically. So grain size fraction can be used to trace paleo current strength, because greater grain sizes are usually transported by higher current speeds. And to be more specific, there's something called the sortable silt proxy, I'm sorry, which means that up to a certain size, clays and fine silt behave cohesively, and as the silt gets coarser, it starts to become cohesionless, and can therefore be sorted very well. And the different organic SST proxies are associated with finer grained, high surface area mineral particles, and may therefore be susceptible to hydrodynamic sorting, which may potentially lead to these temporal decouplings of the original signal. So as for the results of the bulk organic matter characteristics, the organic carbon content of the samples vary between 0.5 and 1%. And surprisingly, there's almost no correlation with water depth, which is usually expected because the organic carbon decreases as it is transported along the continental slope, because it is degraded. Additionally, the OC content is relatively low. And because we are in an upwelling setting, we have oxygen poor conditions, which tend to preserve the organic matter, and organic lateral mixing might also obscure this trend, which is usually observed. Then as for the carbon nitrogen ratio, the values are between 3.4 and 1. Again, there's no correlation with water depth, as it is related to the organic carbon content. But the values suggest that there's no or little input from indigenous sources. We also looked at the delta 13C values, which have a very low variability, but these values reflect the isotopic composition of marine organic matter at this latitude of about -22 per mil, which is in good agreement with the carbon-nitrogen ratio indicating low terrestrial input. Then the 14C ages of the matter show a weak to moderate correlation with water depth, but the relatively large differences of the ages suggest or may indicate input of pre-aged material. As for the sedimentological characteristics, there's a very weak correlation of TOC with mineral surface area, and almost no correlation of 14C age with mineral surface area, which is not in agreement with other data that show that organic carbon is generally increases with mineral surface area. The reason for this may be that with our samples, the grain size is relatively homogeneous. So these data may reflect a small scale trend, and it is very well possible that this distorts the data, and this trend would only be visible over a greater range of grain sizes. As for the sea surface temperatures that we obtained, the values for the UK37 index are between 8 to about 22 degrees. The TEX 86 values range between 15 to 22 degrees, so they are warmer than the ones obtained from the alkanones. And the magnesium-calcium ratio gave us values between 15 and 19 degrees. And if we compare this with the World Ocean Atlas 13, it is apparent that magnesium-calcium derived SST most accurately depicts the mean annual sea surface temperature, whereas UK37 gives a bit colder temperatures, and the TEX 86 index gives a bit warmer temperatures. So the colder bias of the UK37 SST can be explained by some seasonal upwelling during the summer months, where the upwelling currents from come from a northerly direction and can carry material from colder regions further north. The TEX86 ratio is derived from Thormarchiota, as I have mentioned, and these organisms mostly thrive during the summer season. Then, because we have a high nutrient setting, this also influences the TEX86 temperatures towards warmer sea surface temperatures. And we also have an input of warm and nutrient-rich water from the Mediterranean via the Mediterranean outflow water. And within this water mass, we also have a population of deep-dwelling Thormarchiota, which introduce a warmer bias to the TEX86 derived SST, as can be seen from these data points. This is not my data, just to mention this. Yeah, so in conclusion, we can say that we found discrepancies between SST proxies, which can be readily explained by a combination of hydrological and ecological influences. And it would be interesting to kind of look at this in the Benguela upwelling system as well, because here we have a larger variation in TFC content and mineral surface area, which might eliminate some of the problems encountered within the study. And it has also been studied before that we have the 14 sea ages between TOC, alkanones, and foraminifera do not show the same ages, so there are some discrepancies here as well. It would be interesting to see how this plays out with the sea surface temperature values that can be derived from these proxies in this setting. So thank you for your attention.
Thank you, Nicola.
So it's my pleasure to introduce Yan Ma. He did his bachelor in geology and geophysics at the China University of Geosciences, where he continued on to do his PhD in sedimentology and geobiology at a joint PhD program with Peking University and the Massachusetts Institute of Technology under the supervision of Professor Chao-Dung Wu and Roger Summons. And during his PhD, he did part of his work using sediment from the Namibia continental margin, which has been recently accepted at a PNAS journal, and he will be presenting part of his results in this talk. So the floor is yours, Yanma.
Yeah, thanks. So sorry to hear that. Deeply sorry for Chibo. But anyway, thanks Sarah's introduced. And thanks Catherine French. She has report our research background and the results last month. And for this time, I will show more information about our motivations and biomarkers results in Benguela upwelling system. Biomarkers in sediments or rocks can provide valuable information about the communities in aqueous environments. Recognized biomarkers as photosynthetic pigments are mostly produced by anoxygenic phototrophic sulfur bacteria. Different from oxygenetic photosynthesis, these bacteria can utilize the hydrogen sulfide and light to produce the organic matters when the hydrogen sulfide protrude into the fluid zone. Therefore, the detection of these bacteria or biomarkers indicate the existence of the fluid zone Eosinia. Specifically, the ochinol is derived from the purple sulfur bacteria, while the chlorobactin and acerinuretine originate from the green sulfur bacteria. As green sulfur bacteria are strict anaerobes and can utilize slight light. Therefore, in this widely accepted geological model, the occurrence of acerinuretine indicate the fluid zone Eosinia in deep or open oceans, while the occurrence of ochinol indicate the photoexon Eusenia in restricted basins or from microbial mats. Actually, photoexon Eusenia and their related crocodiles in modern environments are serving as modern analogs for ancient strata, which can explain or coevolve with many geological events. In modern environments, the anoxic basins and the furrows has provided good examples for photoexon Eusenia. However, biomarkers, these crocodile biomarkers are really detected in modern open oceans, which has led to the no-analog problem. In modern open ocean environments, the upwelling zones are the ideal locations for detecting the photoexon Eusenia, especially for Benguela. In Benguela upwelling system, there has been noticed that the sulfur eruptions in satellite images in Benguela upwelling system, the sulfur eruptions and the high productivity has protected the anoxic and the sulfurous environment, supporting the anoxygenate photosynthesis by green sulfur bacteria or purple sulfur bacteria. However, using the RC mass, we actually didn't detect any photoexon Eusenia related crocodiles. So we presume these biomarkers would be trapped in organic sulfur compound. Theoretically, the sulfide can easily incorporate into the unsaturated organic molecules, forming the complex organic sulfur compound. For crocodiles, these double bonds in isoprenoid chains can easily get sulfurized during the early diagenesis in water column or recent sediments. However, using the Renyi-Nickel reactions, kind of desulfurization, we could break these carbon and sulfur bonds and release these original hidden biomarkers. So we tested the nonpolar fractions from the sediment extract in Benguela and conducted the desulfurization on polar fractions in sediments extracts. From the GCMAS results, we can see that suboleic alkenes and styrenes and isoprenoid organic sulfur compounds dominate before the desulfurization. However, the biomarkers after desulfurization has shown the contrasting patterns, including the high abundance of phyton, intact alkenes and styrenes and hopens. Based on GCQQ mass, we can see that the neonated styrenes and hopens are features as the biological isomers, such as the alpha-alpha-alpha-R or beta-alpha-alpha styrenes or beta-beta hopens, comparing to their geological isomers. So these neonated biological isomers indicate the sulfurization in Benguela upwelling system happens in very early diagenesis. More important, we detected the photoexon Eusenia related crocodiles. Among all crocodiles, the isorenirete and the beta isorenirete are dominated with small proportions from renitin and renipoprin. Considering the exclusive origination from the green sulfur bacteria, the detection of the isorenirete here indicated the existence of photoexon Eusenia and the existence of the green sulfur bacteria. And interestingly, the isorenirete and the beta isorenirete has shown the strong correlations indicating their similar originations from green sulfur bacteria. While the small proportions of renitin and renipoprin was interpreted as the cyanobacteria sources, according to our recent research from Xinjiang 2020. So to sum up, our research indicate the new findings revealing the green sulfur bacteria and proliferate during the episodic sulfur eruptions, which is very important for microbiology, paleoceanography, and geochemistry. And for following work, we want to conduct down-core samplings for detailed photoexon Eusenia evolutions in Benguela and survey more surface sediments in other upwind system or OMZs. And we could do more applications on radical reactions. Potentially, we also can conduct the DNA detections or some culturing by Diana and MIT. That's all. Thanks.
So, Mr. Rida, I think being a sailor's son has always been a real motivation to get him involved in the world of sea and the ocean. So he's a PhD student in biological oceanography. He's working on ecology and structure of zooplankton population in Mediterranean with biochemical and modeling approaches. His work is a part of oceanographic monitoring of marine environmental undertaken by our National Institute of Fishery Research to study plankton biodiversity and its variability in relationship with the factors of marine environment. He did participate, of course, in RGNO 2019 course, and this course was a good opportunity for him to learn more methods, of course, and skills of analyzing the ecosystem processes through the multidisciplinary studies of upwelling ecosystem. So Rida, it's your turn to present now. Go ahead.
Thank you, Doctor. So hello and welcome everyone. I have the pleasure to present before you my work, which is entitled Effect of Diurnal Vertical Migration of Zooplankton in the Northern Benguela. This work is divided into these categories that you can see on the monitor. So zooplankton dominates the pelagic environment ecosystem and plays a key role in the ecological balance of all ecosystems. Many species of these small animals which drift above the water column display vertical migration due to the hydrologic, physiologic, and trophic factors. However, the diurnal vertical migration or phenomenon of negative phototactic practiced by many species has physiologically and ecologically reasons, namely the action of the light, the nutrition, reproduction, and predation. The Benguela current large marine ecosystem is situated off the west coast of southern Africa, and is one of the 4 eastern boundary upwelling systems in the world. Its high level of primary productivity support an important global reservoir of biodiversity and biomass of zooplankton and fish. So the Benguela region has distinctive bathymetry, hydrology, chemistry, and trophodynamics, which combine to make it one of the most productive ocean area of the world. So the objective of this work are to demonstrate the effect of zooplankton migration and to study the role of zooplankton DVM in the biological pump and carbon export, to demonstrate the brewing layer with an experiment, and to show the migratory behavior of zooplankton using ADCP. So an oceanographic survey was conducted in May 2018 along the Namibian coast at the line 23 degrees south, and a total of 4 zooplankton stations were sampled for my experience. This were done between the day and the night. And the study concerned 2 important phases. The first one is sampling on board Mirabilis, and at each station, the oceanographic parameters like temperature, salinity, and oxygen were measured using the CTD. And zooplankton was collected by vertical holes using a VP2 net of 20 centimeter diameter opening and 2000 micrometer mesh size, equipped with a flow meter. And to count the relative abundance of zooplankton, we use EDCP. So The second phase is done in the laboratory and consists in examining the sample of zooplankton under the binocular stereo microscope. And after that, we just keep the same species that live on the surface at night and deep during the day. And zooplankton species which display DVM were identified by stereo microscope. We found some calanoid, chaetogenate, Euphausiaceae, siphonophores, and salpidae. For data analysis to count the relative abundance of zooplankton, we use EDCP and MATLAB to do statistical analysis. An ADCP is essentially an echo sounder that uses the Doppler effect by transmitting sound at a fixed frequency and listening to the echoes returning from the sound scatter in the water column to calculate relative abundance of zooplankton. And my experiment consists to prove that some zooplankton species go with the DVM, at night go up to the surface, and during the day go down to the deeper layer. And to demonstrate that, we use two aquariums. The first one with zooplankton and the other without zooplankton. For every aquarium, we put two layers of water. The deep with low temperature and high salinity, and the one below with high temperature and lower salinity. And for the species harvested through the Mirabilis research vessel, where we sampled, we just keep the same species that live in the deep during the day and on the surface the night. And to assist the zooplankton migrated, we use the flash of my phone and switch between the bottom and the top of the aquarium. And the end, we use Lipton tea like as a dye to have the color of the layer. And based on the time, there was an increase and decrease in the value of relative abundance of zooplankton in the morning and night time. The yellow to red color variation indicates zooplankton vertical migration. The red color represents the larger value produced by a high density of zooplankton compared to the density that is represented by blue color. We found that the zooplankton migrated to the top at night, staying in the deeper during the day. And for the first experiment without zooplankton, two layers are distinct on 3 hours and 30 minutes. However, for the second experiment with zooplankton, in less than 35 minutes, the two layers were already mixed. So as a conclusion, this general upward downward movement of certain species population is influenced by environmental factors such as sunlight availability, food availability, or dissolved oxygen concentration and temperature. So the zooplankton hides deep during the day to escape its predators and go back to the surface at night to fill by filtering the water filled with the phytoplankton and their reproduction. And this vertical migration of zooplankton population contributes to the mixing of the water and allow their oxygenation as well as the circulation of the nutrients. In addition, this abundant consumption of phytoplankton can sometimes prevent the bloom and avoid asphyxiation of the movement of the environment, and also impose on their predator identical movement. Donc we will have more brewing layer. So for my next experiments, I will try to do with the height and low nutriment layer, aerobic and anaerobic layer, and to identify exactly the species doing the DVM, and more studies on seasonal antigenetic migration, especially on calanoid, on carbon transfer. And before I finish, I want to dedicate this work to the memory of Shibu. My heart left condolences, dear Shibu. Words cannot express my sorrow for your loss. and we belong to God, and to Him we return. Thank you.
So thank you so much, Reda, for this beautiful word about Shibu, and for this nice presentation too from the RGNO 2019 course. And I think your presentation is the last one. So I go back to Kurt.